Technology at College of Natural and Computational Sciences, Hamaya University at Ethiopia. So Sasikumar is presently working as Associate Professor, School of Biological Sciences and Biotechnology, College of Natural and Computational Sciences, Hamaya University, Ethiopia since he completed his undergraduate degree from Vivekananda College, Madurai. He obtained his MSc degree from Government Arts College, Coimbatore. He completed the PhD program in botany from Haryana University, Coimbatore. He has served as associate professor and head of Department of Biotechnology and Department of uh, Environmental Technology and Terpagam University, Coimbatore, and has served as associate professor of biology in Institute of Technology, Asmara, East Africa. His specialization includes bioactivity studies and phytochemical, uh, ethnochemical plant species and fruits, and his current research activities include isolation, identification, characterization of antioxidants, antibacterial constituents. He, he was a senior editorial board member of Research Journal and reviewer of several national and international journals. He has published many research papers both in national and international peer-reviewed journals. He has been granted various fellowships and various projects. I now welcome Dr. James Sasikumar for his talk entitled Biosynthesis of Nanoparticles and Their Applications. Dr. Sasikumar, I hand it over to you.
Thank you. Okay. Thank you, sir, for giving this opportunity uh, to present some research specialization of our team. And I first thank the head of the Department of Botany, PSGR KRISHNAMMAL College, Coimbatore, for giving this opportunity. Actually, I chose this title after seeing the target audience. Most of them, it has been mentioned as research scholars and PG students. So, in that way, I have uh, designed this presentation to diversify their research areas, including this biosynthesis of nanoparticles. So, our topic is biosynthesis of nanoparticles and their applications. For several years, for several decades also, the word nanotechnology has been famous among the researchers and scientists all over the world. And since this is a revolutionary field of manufacturing uh, nano-sized particles by using physical and chemical changes. And this nanotechnology, it deals with the production of nanomaterials and their applications of in different fields. Generally, the nanoscience involves in designing and fabrication of nanoparticles and their applications in different fields. This is a study of reduction of a bulk materials at atomic or molecular level, and this is an interdisciplinary science which includes physics, chemistry, and biological sciences, and still it is a fast growing area of research. And and nowadays the global market of nanotechnology is booming because of its diverse applications in various fields like nanoelectronics, medicine and healthcare sector, energy sector, biotechnology, and information technology. Okay, for the beginners, they should know the basic thing of uh, nanotechnology. That is the definition. Here, the definition of nano is it originated from Latin word called nanus, which means dwarf. And this, if you go for the definition, this nanomaterial, nanoparticle is 1,000 millionth of a meter. That is, 1 nanometer is 10 to the power of 9 minus 9 meter. So the long back in 1959 itself, Nobel laureate called Richard P. Feynman, and he um, narrated about, and he about the miniaturization of the particle, that is the nanonization of the particles. And before his time, people were talking about the chip. After this guy only started using the word miniaturization or nanonization of the bulk materials. Generally, these nanomaterials, they may be zero dimensional, what we are going to discuss this nanoparticles, and two-dimensional nanorods, that is the aggregates, nanorods or nanotubes, and two-dimensional, we know that thin films, thin film physics, it's very famous, and in solid state physics. Okay, and when you take this, the shapes of nanoparticles, they occur in different shapes like spherical, triangular, and whatever we have given, rod-shaped, ellipsoidal, and these shapes, deriving these shapes is based on the reactions. But we are going to discuss those things. And these nanoparticles, when they aggregate, they will build blocks of complex nanostructures like nanochains, nanowires, nanoclusters, and other nano aggregates. You can see in the picture different the nanoparticles. The above one you can see the gold nanoparticles, the wine red color gold red particles, and you can see the size of other biosystems, like you have the cells, microorganisms, and even the functional proteins are also coming under this nanoparticles. If you take the nanosystems, there are four important classes in which the metallic nanoparticles based on gold the gold, silver, even platinum, palladium. So metallic gold particles you can derive from bulk materials of the gold chlorate and silver nanoparticles from silver nitrate. Likewise, there is bimetallic nanosystems, nanoparticles, metal oxides like zinc oxide, titanium silicon oxide, metal oxides. This is and magnetic nanosystems also. And we are going to discuss on metallic nanoparticles. In metallic nanoparticles, there are two very common nanoparticles, gold and silver nanoparticles. Both of them are having unique properties, electronic and optical properties with their surface functionalization. Especially the silver nanoparticles are having high electrical conductivity, thermal conductivity, and they are known for their catalytic activity and antibacterial activity with properties. When you go for a general property of metal nanoparticle, and one of the very important properties is the surface of the atom. The atoms, when they occur as clusters, when the clusters are bigger, the central atom generally it will be surrounded by the other atoms in the next shell, and that is the inner shell. Then inner shell again it will be covered by the upper shell. We know that. So in this case, what happens? The surface will not be larger, like it is reduced one. So the atoms when occur in clusters, they are actually their surface is reduced. So here you have two types like different dispersed clusters and aggregated clusters. Generally, these dispersed clusters have high surface than the aggregate clusters. So this surface generally it is having an impact on a chemical reaction. So when surface area increases, the chemical reaction also, the rate also, it increases. So you can find the high reactivity, the dispersed clusters, they exhibit high reactivity than the aggregate clusters. So surface atom is one of the important characters, one of the important properties of any nanoparticles, because the surface area only determines the application, the property of what's called the nanoparticles. And we have some more other parties like high surface to volume. In addition, these nanoparticles have lower melting points than specific optical properties, mechanical strengths, and specific magnetizations. Because of these properties only, they are attractive in various industrial applications. And optical property is one of the important attractions of a nanoparticle. For instance, gold generally, we know the color of the normal bulk material gold, but once if it is reduced, then it will become a wine red color. And silver nanoparticle, it will become yellowish gray color, and platinum and palladium, after reduction, they will exhibit black color. Okay, so now we have seen a small introduction for nanoparticles and how to fabricate the nanoparticle, how to synthesize the nanoparticles, the methods of synthesis of nanoparticles. There are three important methods: physical method, biological methods. In physical method, generally this is called a top-down approach. Top-down approach in the sense an external force is given to the solid. A bang is external force is given to the solid. So then the solid material, the bulk material will be broken down into smaller particles. This is top-down approach. From a bulk material, that is a solid material, is broken down to get smaller nanoparticles. So actually here there are several techniques, there are several methods like grinding, crushing, cutting, degradation, homogenization, disruption, and we need not to go in depth of this physical method since we discuss on biological synthesis. And what are the limitations of this physical method? This physical method, this top-down approach, will produce large sizes of nanoparticles and diameter and volume, and there is surface defects also. This method of pulverizing the solid material into nano materials will cause contamination. Moreover, it's expensive and time consuming. And this is a chemical preparation method, unlike physical method, which is top-down method. This is a bottom-up approach. Bottom approach actually, it's the particle reduction. It starts from the atoms itself by atomic transformations and molecular condensations. There will be reduction of the bulk material into nanoparticles. So in this method, in this chemical method, we have a gaseous phase method and liquid phase method. For gaseous phase method, we follow chemical vapor deposition method, and for liquid phase, we have physical vapor deposition method. I think here also we did not go in depth because there are several methods in chemical, and I have given some examples for reducing agents, reducing and even stabilizing agent also. And here synthetic chemicals are used as a reducing agent because the bulk material should be reduced into nanoparticles. So in this case, borohydride, sodium borohydride, even citric acid, ascorbic acid, and these are some elemental hydrogen, and these are used as reducing agent as well as the stabilizing agent. The reducing agent, they will donate an hydroxyl, an electron to the bulk material, and it will be reduced into nanoparticles, and that will be stabilized by these polymers. And what is the disadvantage? What is the limitation here? In chemical preparation, due to the usage of these chemicals, they may cause pollution in terrestrial and aquatic life forms, and it will lead to accumulation of these pollutants in the environment. So because of the limitations of physical and chemical methods, nowadays people have concentrated on green synthesis, that is biosynthesis of metal nanoparticles with the help of plant extracts and with the help of microbes, and this is also a bottom-up approach. And this is a picture, an illustration for reduction of nanoparticles of plant extracts, organic microbes, and algae, mushrooms, and seaweeds. And here you can see the metallic salts, the metallic ions reduced into metallic nanoparticles, that is zero valent metallic nanoparticles. So it's a zero dimensional one, zero valent metallic nanoparticle. And in plant extract, generally we are going to discuss those. And so there are reducing agents like cofactor. We are going to discuss this also. Okay, so now let us discuss biological nanofabrication, that is biosynthesis of nanomaterials, or bioreduction, or you can say green synthesis of nanoparticles. And here we use plant extracts. Plant extracts, they can act as reducing and stabilizing agent, because after reduction of the materials, after synthesis of the nanoparticle, it will be finally you will get a stable metal nanoparticles. So what are the advantages over those physical and chemical methods? Because we have seen that they cause pollution, and these, they are eco-friendly, less expensive, and scaling up process is a very easy one to synthesize a high number of nanoparticles, and no need of high temperature, pressure, and you will see the combined effect or the synergistic potential of nanoparticles and bioactive components in any other applications, in any other bioactivity studies. So how to synthesize these nanoparticles with the help of plant extract and microbes? So here there are three phases of nanoparticle synthesis. The first one is activation phase. In this phase, when you add plant extraction, when you mix plant extract with the metal salts, the metal salts will be reduced by the compounds of the plant extracts, and there will be a nucleation of reduced metal. Nucleation, actually, there is a formation of a crystal. That is not, it's not a growth. The growth is different. Nucleation is different. There will be homogeneous nucleation. Just the start point of what's called the crystal structure, the growth. And this is the first phase. The second phase, there will be aggregation of nanoparticles, small nanoparticles. When you prolong this growth phase, when the reaction time is exceeded, in those cases, what will happen? There will be aggregation of these nanoparticles into nanorods and nanotubes. So we have a certain reaction time for the growth phase. And finally, termination phase, and here this final shape of the nanoparticles are determined. This is determined in the termination phase, that is the conformational change. The shape of the nanoparticles is determined in the termination phase by the stabilizing agent. Okay, so after this reaction, what will happen? How do you observe the results? Initially, the primary indicator of the nanoparticle synthesis is the color change. Color change will give you the primary indication of nanoparticle synthesis. So in this, for example, if you take silver nanoparticle biosynthesis, it will give a dark brown color. So when you see the dark brown color, you can say as it is silver synthesis of silver nanoparticles. Sometimes what will happen, there won't be any change in the color of the reaction mixture even after reaction. So in that case, it may be because of the acidic medium. Mostly because of that, we sometimes add sodium hydroxide. Most of the time, the alkaline medium favors the synthesis of nanoparticles. Okay, so I told you that plant extract can act as both reducing agent and capping agents also. What is the need of the capping agents? Most of the time, in other chemical methods and other methods, people have been using commercial surfactants, some polymers, but they cause they do not degrade. Actually, they do not degrade, and they cause hazardous effect to the system. So we go for biomolecules for as capping agents. So here plant extract, they act as both reducing and capping agents for fine control of nanoparticle synthesis, which will control the morphology, size, and surface protection for a better function and stability. So for a better function and better stability, you have to control the morphology and size and surface protection. For that, you need these capping agents. So here, this is a natural plant capping agent, and this is a single pot reaction. Like the plant acts as both reducing and stabilizing agent. So this is a single pot reaction. Okay, so after this, this is the reaction mixture, and you will get the color, and further you will go for characterization. And okay, what are the factors that are affecting the nanoparticle synthesis? One of the factors is extract concentration. Plant extract, generally you can extract from any parts of the plant, like seed, leaf, stem, flower, any even callus also. You can take any part of the plant for the extraction. And since ours is a nano formulation, a minimum quantity of extract is required. A minimum quantity of extract is required for a fine synthesis of nanoparticles. And when we go for metal salt concentration, this is very important part, because when you add more metal salt, but the synthesis of nanoparticles may be hindered. So in this case, we need a very minute amount of, a precise amount of metal salt concentration is required in this case. Okay. Then the next important factor is reaction temperature. Temperature, when the reaction mixture is heated, generally these electrons from the secondary metabolites of the plant extracts and from the metal ions are excited. They are oscillating. This excitation will cause what's called, there will be homogeneous nucleation in the nuclei of the metal ion, and oscillation. There is a band called surface plasmon resonance. So the increasing temperature will cause the immediate homogeneous nucleation and SPR, that is surface plasmon resonance. I will explain you later. So because of this, you will get a smaller, when temperature increases, you will get a smaller nanoparticle with stability. Okay, reaction time also plays an important role here, but some studies differ in their results. Like when reaction time increases, there will be reduction of the growth of the nanoparticles, but not the size of the nanoparticle. Size of the nanoparticle will be smaller when you increase the reaction time, but you will avoid getting the larger nanoparticles when you increase the reaction time. And the pH, what is the effect of pH on this reaction? I've already told you that actually this pH will determine the capping and stability of the nanoparticles. And I told you that in acidic pH, there may not be any color change. So alkaline pH will favor for color change for any nanoparticles. But when pH increases, sometimes you will get a spherical, that is an ultimate shape of nanoparticles. You will get spherical nanoparticles when the pH increases. So these are the factors that are affecting the nanoparticle synthesis. Okay, so in the plant extracts, without any, there are phytocomponents. With the help of these phytocomponents only, these metal ions are reduced into metal nanoparticles. We have several compounds like flavonoids, terpenoids, vitamins, catechins, tannins, and pigments, plant pigments. So let us discuss the flavonoids, how the flavonoids are involved in metallic nanoparticle biosynthesis. If you see the flavonoid biosynthetic pathway, that is accumulation acid pathway, these flavonoids have come across several chains, several network in which these flavonoids have been polyhydroxylated. All flavonoids have been polyhydroxylated with oxone to hydroxyl group along with flavonoid catechol moiety. And this hydroxyl group, see, they have the power of bond breaking that is found in the catechol moiety, which will reduce the metal ions. So during this reduction of metal ions by hydroxyl groups, there will be a transition of flavonoids, that is keto-enol tautomerization, keto to enol, enol to keto. During this transition, there will be release of what's called reactive hydrogen atoms. Once this hydroxyl ion, it is released, the hydroxyl ion is, the hydroxyl group is converted into carbonyl groups. The hydroxyl group will reduce the metal salt. Then after conversion, the carbonyl group will stabilize the nanoparticles. So this is the mechanism of biosynthesis by flavonoids. And you can see there are some functional groups. This can be observed with the help of Fourier transform infrared spectroscopy. And I can give some examples of flavonoids that can reduce the metals, metal salts into nanoparticles. For example, naringenin. Naringenin is when you see the pathway of flavonoid synthesis, this is a transitional molecule. Its precursor is naringin, and from naringenin only there will be side chains to produce other flavonoids like flavonols, flavonones, anthocyanins, and anthocyanidins. So naringin is the you can consider as the first compound of flavonoids here. So in addition to this, naringin, diasmin, hesperidin, also they can reduce this metal salts into metal nanoparticles. Okay, so how could you prove it? Sometimes we need a correlation to prove the reduction capacity of the flavonoids. When you consider the antioxidant activity of the flavonoids, here the antioxidant activity, when they scavenge hydroxyl ion or superoxide ions or nitric oxide or hydrogen peroxide, they donate an hydrogen atom from their hydroxyl group, and they will scavenge the free radical. The same manner here also, the flavonoids, they donate the reactive hydrogen atoms from their hydroxyl group, and they will reduce the ions into the metal ions into the nanoparticles. So there is a linear relationship between this antioxidant activity and the reducing metal salts by these flavonoids. So this is phenolics. Actually, it's a major group of compounds, and here there are some examples. We have caffeic acid, gallic acid, protocatechic acid. In addition to this, there are several phenolic acids are involved in biosynthesis of nanoparticles. And here also there is a complex for transition complex will be formed with metal ions with what's called phenolic hydroxyl groups. So the next group of compounds that are involved in biosynthesis is terpenoids. Terpenoids or isoprene compounds is called isoprenoids. In terpenoids, generally we can see the essential oils. We have monoterpenoids, di, tri, and sesquiterpenoids, and they are responsible for the aroma of the plants. And they have the functional alcohols, functional ketones, aldehydes, and lactone. And from this, the hydroxyl groups will release their reactive hydrogen atoms to reduce the metallic ions into metallic nanoparticles. So in addition to this, even low and high molecular weight proteins are also involved. And after reaction with the metal ions, after synthesizing metals, you can separate these proteins using electrophoresis. And there have been several studies on this one. And here actually in proteins, because of peptide bonds and because in the presence of some specific amino acids, free amino acids like tyrosine and phenylalanine. And in addition, the exposed regions like the exposed disulfide bridges of proteins and thiol groups, they are responsible for reducing and stabilizing the nanoparticles. Generally, the proteins the metal ions are interacted with the proteins peptides, one protein, bridges, protein thiol groups, whatever they are interacted with the help of electrostatic interactions. Then after electrostatic interaction, there will be reduction of ions by the proteins. So here also the proteins act as capping agent, stabilizing agent. Okay, microbial synthesis. So we have seen the how plant extract can reduce the metal salts into nanoparticles. Here, this is microbial synthesis, and here the microbes, from the microbes, the extracellular enzymes, sugars, and proteins, they are capable of reducing the metal ions into nanoparticles. And microbes, they have some speciality, their behavior and interaction differently with different metal ions. And this, their interaction with the different metal ions and their bioprocessing activities, and in addition to that, the environmental pH and temperature, everything, all these factors, they determine the formation of nanoparticles with a particular shape and morphology. Because we need a particular size. The size should not exceed, the nanoparticle should not be larger. So we need a specific size. So that size and the shape, the conformation, the morphology should be determined by the interaction of microorganisms with the metal ions and their biochemical processing, and even the environmental pH and the temperature. And in addition to these enzymes and sugars, there are some other secondary metabolites like anthraquinones, hydroquinones, and naphthoquinones. They also play an important role in biosynthesis of nanoparticles. And I told you that there is a cofactor NADH. It acts as electron carrier, it's in collector, and an electron is transferred to reduce the metal ions with the help of NADH independent reductase. And we know that the microorganisms generally, they can tolerate high level of metal ion concentrations. Like we have already studied that the bioabsorption and bioaccumulation of heavy metals. So this property of microorganisms helps in reducing this metal ion concentration also into metal nanoparticles. And in addition to that one, we have some other reducing agents also. Algae, there have been several studies on algae, because of algae generally we use the environmental biotechnology, we use it for bioremediation. Since it is used for bioremediation to bio-leach these heavy metals, that could be used for synthesis of nanoparticles also. And in addition to that one, we can go for waste material also for the synthesis of nanoparticles, especially from food waste and food industry waste, because this food generally they contain the cellulose, hemicellulose, the pectin, and the biodegradable polysaccharides on their cell wall. So these components found in the cell wall of this food will help to reduce the metallic ions into metallic nanoparticles. And in addition to that, there are biodegradable agro waste also could be used for the synthesis of these nanoparticles. Okay, so after synthesis of nanoparticles, we had the reaction with the metallic ions and the plant extracts or microbe, and after the reaction, there is synthesis of nanoparticles. Then that nanoparticle should be subjected for characterization. Yeah, here we have different methods. Let us see one by one that UV visible spectroscopy. Mostly this UV visible spectroscopy is used to study the optical properties of the nanoparticles. We know that the nanoparticles, when it is subjected in UV spectrophotometer, it will show the different characteristic colors. And during that time, these nanoparticles will absorb this electromagnetic radiation. During that time, there will be oscillation of the electrons on the surface of the metal. So because of this interaction, there will be a coherent oscillation, a covalent oscillation of the electrons on the surface of the nanoparticle, the metal nanoparticle. So these resonances are known as surface plasmon resonance, which occurs in the case of nanoparticles. So when after during characterization, you will read the peak of the SPR, that is surface plasmon resonance, the width, the SPR width, the SPR band will be read in UV spec, and so sometimes it will be 500 nanometer or 400 nanometer. So that nanometer, that peak will be read with the help of UV spectroscopy by reading the band of surface plasmon resonance. Okay, this is the initial what's called characterization of the nanoparticle synthesized, and then transmission electron microscopy. This is like just like a slide projector, but they transmit the beam of the electron and directly to the phosphor screen, and from there you will get the final image formation, and this will give you the information on topography of the nanoparticles, the morphology, the shape, size, and all, and the crystallography of the composition and crystallography of the samples. This is transmission electron microscopy, we call it. And this is SEM, scanning electron microscopy. This is somewhat different from the SEM. And here you can determine the size, shape, and surface morphology of the nanoparticles. And here there will be an electron beam which will be scattered like secondary electrons and scattered electrons, and that will be collected by the detector. Finally, the image will be analyzed for their shape, size, or surface morphology. Okay, then the X-ray diffraction is called XRD, and this is one of the important techniques used for the characterization of nanoparticles. Generally, it will provide crystalline structure and these latest parameters and crystalline grain size. So X-ray diffraction, it generally shows the crystal phase of the nanoparticles. So this is the way the powder sample, when you keep on the holder, the X-rays of this wavelength is recorded using a meter called goniometer. Generally, this XRD will give nanoparticles a broad peak. So by this, in addition to TEM and SEM, X-ray crystallography also should be used for characterization of nanoparticles. And this Fourier transform informed spectroscopy, this is for surface chemistry of the nanoparticles. I have already told about the functional groups. So some functional groups will be identified with the vibration of chemical bonds in the molecule with the help of this FDIR. So the functional groups attached in the nanoparticles, they have different FDR pattern in plant extracts and different FDR in resulting nanoparticles. So the functional groups that are found in the nanoparticles, then that are responsible for the reduction of metal ions into nanoparticles or read in FDIR. Okay, so we have seen how to after synthesis, how to characterize the nanoparticles. And there are several applications. There are a number of applications of this. I'm going to give you very few. For example, these nanomaterials, they can be used for manufacturing sunscreen lotions to save us, to protect us from UV radiation for a prolonged period of time. So for example, we have nano titanium oxide. This is an metal oxide nanoparticle. This is not a normal metallic nanoparticle. This is a metal oxide nanoparticle, which is included in sunscreen lotion manufactured. And the next application is bioimaging. Generally, the large-scale materials that are used for bioimaging, or they are photo bleaching. They can be degraded easily. So they have, because of their disadvantages of this photo bleaching and all, the bioimaging by nanoparticles or nowadays is common. because they are photostable. Because if you see the nanoscale gold, they can appear red or purple color, so they can accumulate on the cancerous cells and the tumor, and you will get a precise imaging, and finally you will avoid the harming of the healthy cells. So and you can distinguish the healthy cells from the cancer cells with the help of the antibodies joined to the nanoparticles, and you have a specific color also, so that your the cancerous or tumorous cells will be easily imagined. So this is one of the applications, and the next one is antibacterial activity. The nanoparticles, they have several activities: antibacterial activities, antioxidant activities, anti-cancer activities also. But in this antibacterial activity, this is actually depends on the size and surface area of the nanoparticles. So with the help of the size and the high surface area, these nanoparticles will suppress, they will suppress some enzymes that are responsible for cell wall synthesis. So automatically, once the enzymes are suppressed, then there is no cell wall synthesis of the bacteria, so the bacteria will die. And since they are very small, with a high surface area, they can adhere to any microbes, and they can break the bonds. Finally, they can enter into the what's called bacterial cell by penetrating the peptidoglycan layer. And mostly the spherical shaped nanoparticles, they have some compatibility with this bacteria, so they can easily interact with the cell walls, and even if it is small size, there will be high penetration. So because of this, their properties, their size, their shape, and their high surface area, they are able to inhibit any bacterial pathogen. So I told you that other than this antibacterial activity, we have and an antifungal activity, antioxidant by scavenging the free radicals, and anti-cancer activity also. Okay, so but actually most of the nanoparticles, nanotechnological studies have shown their applications in drug delivery, because they have the advantages of the microparticles, because generally the therapeutic drugs are having the poor solubility. Because of the poor solubility in the solvent, they cannot efficiently be absorbed by the body. So once if it is not absorbed, it will not be effective. So what will you do? Generally, you will increase the dosage of the therapeutic drugs. So automatically there will be side effects. So when you go for nanonization of the drug, you will have the nanoparticles have the relative high surface area. So automatically there will be an increased exposure to the solvent. Then the solubility will be enhanced. So the body will absorb it perfectly. So here there is no need of giving, high dosage. The reduced dosage is enough. And targeting the delivery. Generally, these when you use chemotherapeutic normal agents, there will be a poor compliance by the patients. So the targeting the what's called delivery of this cells, the molecules, it's a challenge now. So when we use the gold nanoparticles or silver nanoparticles, by both any active targeting as passive targeting, this both even surface and inside cells also, they can interact with the nanoparticles, will interact with the biomolecules because of their size. Because of their size, they will interact with the biomolecules. So the targeting will be very easier for any therapeutic. Even you can use 10 to 100 nanometers of different shapes and size of gold, nickel, and silver, iron metal nanoparticles for this drug delivery system, because of their surface area, because of their smaller size, because of their solubility, there will be better absorption. So the delivery will be perfect. So in addition to these nanoparticles, you can use some encapsulated drugs in some nano-sized materials. There are several nano-sized materials, the dendrimers, hollow polymer capsule, nano shells, even we have fullerenes. There are a number of nano-sized materials that are used to encapsulate the drugs to the target. This is the polymeric, and this is a nanoparticle with you can see the drug. You can see the drug, and this is the ligand. This is the targeting ligand, the targeting molecule, and this is our nanoparticle. And some more applications for the manufacture of some biosensors like DNA impedance biosensor for the detection of cancer and the detection of flu virus. We can go for integration of DNA and other nucleic acids with nanoparticles. So that will produce some composite nanomaterials. These composite nanomaterials, because of two components, they can exhibit synergistic activity. So because of this, this principle, our scientists have developed DNA impedance biosensor for early detection of the cancer and the flu virus. In addition, in food sectors also, they have a lot of, the nanoparticles have a lot of applications, especially in pesticide delivery systems using nano encapsulation and biosensors to detect food pathogens and to quantify, even for quantification of these food pathogens also, and to preserve fruits, edible thin films. Thin films, I told you, it's a two-dimensional nanoparticles. So these are all some important applications. There are several applications. I have given you very few applications for you. Okay, this is actually our team, what we are doing now. We have started the work on green synthesis of silver nanoparticles using the Ethiopian medicinal plant extracts and their activity against Helicobacter pylori, which is causing ulcer. This is a predominant found in some Ethiopian, this African countries, because what's called the ulcer. So we have tried to collect plants that are curing, that are traditionally used to cure or treat the ulcer, and we have collected and we have synthesized the nanoparticles, and we have done some initial characterization, but this time this COVID-19 has invaded and it has crippled our hands to stop this work temporarily. Okay, so in the future, it's very simple. Like we know that the nanoparticle, the biosynthesized nanoparticles are eco-friendly. It's less expensive, and both plants and microbes, they are able to synthesize the nanoparticles for several applications. And the problem here is the identification of bioactive compounds, because it is a time-consuming one. It's a very complex procedure. So what happened there? Further progress will be delayed because of this identification of bioactive compounds. So we need to work. Some more studies, extensive studies, are required to find out the exact role of the biocomponents for the synthesis, stabilization of the nanoparticles. And phytocomponents should be characterized that are responsible for nanoparticle formation. So the identification and characterization of these bioactive compounds that are responsible for the reduction of metal ions to nanoparticles. And there are several methods. There are so many methods we have used to synthesize nanoparticles and for characterization also. So we should improve the reproducibility of this method. And there are a number of methods, mechanisms are involved in the formation of nanoparticle from the metal ions. And from the different plant, you will get different sizes of particles, different shapes of the particle, and different colors of the particles. So, and different reaction time, different temperature gives different sizes and shapes of what's called the nanoparticles. So we need to develop, identify or detect or elucidate a clear and precise mechanism of how the nanoparticles are formed by biogenic methods, because the morphology of the size and morphology of the nanoparticles only will determine their applications. So a clear mechanism, a precise mechanism should be elucidated. And in future, I think we don't have a large scale production of nanoparticles in India. So we could go for a large scale production using the phyto and bio-nano fabrication. And this presentation on biosynthesis of nanoparticles I have prepared only for most of the students who are involved in research. They are research scholars and the PG students. So thank you so much for giving this opportunity. Thank you.
Thank you, Dr. Sasikumar. That was very comprehensively covered. I could see that you started with the formation, characterization, then application. You have very well covered the complete topic. It was very understandable to all of us. We all come from different backgrounds. Now, I would like you to take up a few questions. I will be reading out the questions from the chat box. So the first question is from Subhadarshini Farida. She is asking that is there any side effects when nanoparticles are used as a therapeutic agent?
Yeah, there are some studies of these nanoparticles from the bloodstreams. When they penetrate into some organs in the body, they will cause some toxicity, like lungs and liver and even lungs also. There have been several studies on that one.
All right. So must be depending on the source of nanoparticles. Like it might penetrate into the organs also. Yeah. So a detailed study is required before actually we prescribe such kind of nanoparticles for the
nanoparticles. Yeah. Some toxicity studies are required.
Right. The next question comes from Deepika Patnaik. She's asking whether which group of nanoparticles can facilitate drug transport across blood-brain barrier. So, can the blood-brain barrier be crossed by nanoparticles?
What measures have been taken? No, I will read. Is it readable?
Yeah, Lipika Patnaik, she asking which group of nanoparticles can facilitate drug transport across blood-brain barrier?
Okay. I haven't come across any specific nanoparticles because we have started with the basic synthesis and activity against the bacteria and fungus. So we haven't come across such nanoparticles.
All right. Next question comes from Dimpi Das. She says, "Sir, can you please give some examples of the microbes or plants that are used in synthesis of nanoparticles?"
Even Aeromonas, we have started. We have initialized in some of our laboratories with Aeromonas hydrophila, and some Pseudomonas species. But we have started recently with Aeromonas.
All right. The second part of the same question is that if a new plant or microbe is to be used for synthesis, do we need to check for particular characteristics?
Characteristics of?
The question is slightly incomplete, but I presume that she wants to know that what characteristics should we look for when we are using a new plant or a new microbe?
New plant, okay. It's not a new plant or old plant, whatever it is. When you extract a plant, generally we go for a literature for the presence of compounds and all. But all the plants, they must be having the secondary metabolites, the phytochemicals that are responsible for the reduction. So there is no need of search for a special character from a new plant. So any plant, there are phytochemicals. But any new plant, whatever the study you do, you will search their background information. So that background information is sufficient for carrying out this type of work. So there is no special characters, special properties required for synthesis of nanoparticles from this new plant.
Okay. One thing more I wanted to ask that you said that while characterizing a nanoparticle by FTIR, you come to know about the various functional groups present on the nanoparticles. So,
Yeah.
what kind of functional groups should ideally be present on nanoparticles? What is the preferable functional group that should be present on a nanoparticle?
Yeah, mostly the hydroxyl groups and the other groups that are the major groups of flavonoids or any compounds. Mostly the hydroxyl groups and other what's called COH groups and other groups that are responsible for reduction of the biosynthesis.
I could see that you have been involved in a project in Ethiopian medicinal plant for the
Yeah,
formation of nanoparticles. What
yeah,
do you
yeah,
think is the potential of India if we see the
yeah.
Indian biodiversity? So
Yeah,
is it
in India, actually, yeah, but in India, our culture is different. Our traditional practices are different. Here, the traditional practices are different. But to any cost, plants in both cases, maybe the species differ. Otherwise, the potential is the same. So in India, also, we have a lot of traditional medicines, a lot of I mean, tribal medicine. So we can use medicinal plants of India for synthesis of these things.
I believe that this technology was used since ages in India, but only now
Yeah, yeah.
we have characterized it with the scientific
Yes,
name.
yes, yeah. Because for several decades, I have seen several reports from long back. But now only we are getting this awareness to synthesize, and we are going for the new techniques, new mechanisms, and characterization. Because very recently, only most of our institutions they received the FTIR, the X-ray, or TEM and SEM and all. So characterization is still it is difficult for many people. Even in Ethiopia, uh, searching these instruments is very difficult. In one or two institutes only we have this X-ray, XRD, and then SEM and TEM, even one FTIR. So there is difficulty in characterization because of these problems.
Yeah. Otherwise, I think you would agree that making of nanoparticles characterization is difficult. I understand because it needs sophisticated instruments. But
Yeah,
making of
yeah.
nanoparticles, that is not an expensive process. I think you would agree with
Yes,
me.
yeah. Sometimes the silver nitrate or gold uh, these particles maybe sometimes it's expensive, but it's affordable. I think it is affordable, and from this we can go for synthesis, and we would go for what's called biological activity studies. That would help the students, but it's possible to go for UV spectroscopy as the initial characterization of the nanoparticles. So we can go for UV spectroscopies for the imaging that what's called the peak, the SPR peak.
Right. Right. So, I would, if there are no more questions from the participants, I would consider that there are no more questions and you have understood.
There are few questions in the chat box.
Yeah. Just a moment. I think Rahana Moyuddin, she is asking, did nanoparticles in drugs affect liver?
Yeah, that I already answered. There is some toxicity of these nanoparticles on liver and lungs also. There are some reports. I've already answered for this one.
Right, Kajari Das, she's asking that what measures are being taken to evaluate the level of environmental pollution generated by nanoparticles released to the nature.
I've said that these nanoparticles generally they never cause any pollution for the environment, except the chemicals released during when we use chemical methods. Otherwise, this is eco-friendly. The biosynthesis of nanosynthesis, I mean, particles is eco-friendly.
Right. I can see a question from Radha. She is asking that can nanoparticles be used in sunscreen, cross the skin barrier, and enter the body? I think you've answered this before. she's
Yeah, I have given an example, titanium oxide based sunscreen, but I don't think it will enter. This is I have given as an example only. I have not, I don't have a good idea of that.
right. Bhagyalakshmi is asking that whether nanoparticles help in photosynthesis also.
Pardon?
She's asking whether nanoparticles can help in photosynthesis also.
Uh, I have not
Yeah,
gone
that's a
thoroughly
new idea.
in such. Yeah.
That's quite innovative thought,
Yeah, it's a
Dr.
good,
Sasi.
it's very good thought, good thought. It is a mind-blowing question, that one.
Yeah,
That we should interrogate. We should interrogate on such aspects.
yeah. Another one is from Sujata Maiti Chaudhary. She's asking, is docking study possible for biogenic nanoparticles?
I hope so, but I have never seen any studies on this one. Maybe it's possible, maybe. Because we have
Right.
started this very recently in these 2 years only. So we have never thought about the stocking studies. So, and I haven't come across much, I mean, any studies on docking. Maybe it is possible if.
Right. Dimpy Das is curious to know that can metal nanoparticles induce metal allergies?
Metal allergies, I don't think so. There is no reports on that one. There is no report on, because we use generally this silver, gold, and platinum. Generally, gold won't make much allergies for us, right? So I don't think silver and gold. So, when it comes to a nano size, it will be safer. So
Yeah,
there is no problem for
right.
this
The rest of the
allergy.
comments, most of the participants are appreciating your simplicity and the
So
beautifully delivered talk, and they have
yeah, thank
sent us.
you so much. Because here there was some riots and violence because of the political issues. So there was no internet, and I prepared in a short time, very short time, to deliver it to the targeted research scholars and the students. So somehow I think I have given the good information to the students and the other audience.
Yes, I agree, Dr. Sasi. This was actually this is a very upcoming field, and we in this FDP program, this is one of our intention that all of our faculty members should be knowing about the potential of this new field, and they can
Yes,
also
sir.
venture into this new topic, and it has tremendous potential looking at the biodiversity of India.
Yes, sir.
Once again,
Yes.
yeah. Once
Thank
again,
you, Dr. Raj.
thanks, Dr. Sasikumar Mahalingam, to be here with us for taking time out of his busy schedule. And thank you,
Thank you, sir.
participants, for listening us patiently. I thanks once again, Dr. Sasi and Dr.
Thank
Shreya.
you, Dr. Rajkumar.
Right.
Thank you so much. Thank you, all of
Uh,
you.
with that, we come to the end of the session, and we look forward to see you tomorrow at 11. Thank
Thank you,
you.
Dr. Shashi. Thank you.
Thank you, madam. Thank you so much.
Thank you, Rajiv.